Good morning students, welcome back to our subject the biology. In first chapter nutrition, the food supply system. In previous class we have discussed that autotrophic nutrition and photosynthesis. In that photosynthesis finally we have learned the photosynthesis equation which is derived by the Robert Hills. It is a balanced equation. It is known that plant synthesize carbohydrates, the smaller, simpler ones first, and from them the more complex ones like starch and cellulose. Plants are capable of and synthesize all other compounds like proteins, fat, etc. Animals are not capable of carbohydrate and they are dependent on the plants. Can you state that photosynthesis is the basic energy resources for most of the living world? As obviously, here the photosynthesis is the basic energy resources for the world. Okay, let us discuss study how the plants prepare carbohydrate through the photosynthesis. Before going to enter this topic, we have to conduct an activity. Okay, activity that is the presence of starch. Okay, the presence of starch in leaves. Okay, dear students, we have to know the some several steps how to conduct activity in the laboratory and how to conduct a activity and what are the procedure and what steps we have to follow and what precaution we have to take in lab. So before this activity we have to know this here. First of all, in conducting the lab activity we have to majorly follow the 5 steps. First one, aim, and second one is the required material, and third one is the procedure, and fourth one is the observation. Finally, result. Okay, it's very simple. Aim. Aim is nothing but whatever they are given in the topic. That is it. Here what they are mentioned, presence of starch in leaves. Here, to know the presence of starch in leaves, that is aim. Required materials. Required materials means whatever the materials required for the activity, that are the required material. Just you have to write down. Next one, procedure. The process. How to conduct the activity. The next one is observation. Observation nothing but here. While doing experiment or after the experiment, we have to observe changes. And next finally that is result. Result okay, whatever we have observed in the doing activity, by doing activity, finally we get something that is called or that is a result. Okay, yes. See here, first one is you have to take the required materials. For first one is a beaker and boiling test tube, boiling water and leaf, methylated spirit and Bunsen burner and tripod stand. Okay, and iodine solution and petri dish. Okay, first of all, let us take a fresh, soft and thin leaf which is well exposed to the sunlight. Okay, and boil it in a beaker. After that, you collect the leaf and again you have to place in the test tube which is contains methylated spirit. Okay, what will happen when you place in the methylated spirit? Oh, it means when you place a boiling leaf in a methylated spirit, the chlorophyll pigment will remove from the leaf. That's the reason. Next, after boiling, and you have to boil again it in the boiling water, and you have to collect the leaf from the boiling tube with the help of brush and place in the petri dish and petri dish. Next time you have to take iodine solution and drop on the leaf, boiled leaf, 2 or 3 drops. That is a procedure. Observation. Observation, nothing. In next observation is here the leaf is converted into blue black color when we add iodine solution to it. It means the leaf can contains starch. Starch. This is our